The final unit in our Evo-Devo section is going to talk about gene duplication. And gene duplication, it turns out, is a critical evolutionary process and is really fundamental to understanding the development of animals, plants, development of all organisms. But in addition, it applies beyond development also. But we're just focusing on the developmental aspect here today. So as we begin to describe and define the different genes that we find in organisms, we recognize that many of them look very similar. And I'm not just talking that, there's a gene in a mouse that looks similar to one in a fruit fly, or there's a gene in a human that looks similar to one in a mouse. That's true, and those do occur. But in addition, we found that genes within the same organism, so a gene in a mouse and another gene in the mouse, often looked very similar to one another and sometimes even had very close related functions. Or they looked similar but had somewhat divergent functions. And so this led us to understand this really critical process for where new genes come from. So essentially, this is a little bit of a simplification, but essentially there are two ways that we can get a new gene. And now I'm not talking about genes mutating and changing over time, because that's just the same old gene that's then changed. But what I mean is, how do we get more genes? More genes than we had before. And so number one, they can evolve de novo, which means we have non-coding DNA. You can have mutations that occur that create a promoter and then have a start codon, and so that it can happen. Now that's a really, really long shot. It has happened, certainly happened early in the history of life on Earth, but since then that's probably a much, much less important source for new genes. Much more importantly, we take the genes that are already present in organisms and we have mutations that cause you to have extra copies of those genes. So a gene duplication mutation makes an extra copy of a gene or a set of genes in the genome. So whereas once we had one copy, we might end up with two. Or we have 4 genes that were next to each other and then we copy all 4 of those genes, so we have now 8 total genes instead of just the 4 that we started with. And so gene duplication events are mutations that increase the genome size of an organism. They can occur from non-disjunction events or from crossing over, from genome duplication when the entire genome is duplicated, and there are a number of others, and those are not real critical. But once we have a duplicated gene, it gives us a new form and a new way to understand the evolution of organisms. So it turns out that there are actually 3 types of homology at the genetic level, at the level of a gene. Now what we normally think of as homology at the gene level, we will call orthology. Ortho means straight. So this is a relationship of two genes that can be traced straight back to the speciation event. So for instance, you have an alpha hemoglobin gene and a chimpanzee has an alpha hemoglobin gene. Those can be traced back to a single alpha hemoglobin gene in a common ancestor. So number 4 and number 5 would be orthologous. Okay? However, when a gene duplicates, when there are 2 copies of it, they're still related to one another because they can still be traced back to a common ancestral gene. That's still homology, but it's a slightly different version of homology. So for instance, you have an alpha hemoglobin gene and a beta hemoglobin gene. If I compared your alpha hemoglobin to a chimpanzee's or another primate's beta hemoglobin gene, I would see similarities. There's similarities in structure and even some maintain similarities in function, although they've diverged somewhat to allow them to do a little bit of a separate job. They work together to help carry oxygen and carbon dioxide in your body. But if I compared them, we would see those similarities at the genetic level. Yet, when we trace them back to the common ancestor, you would still have 2 separate genes. So gene 5 and gene 3, notice, are still, there's the common ancestor of gene 5, there's the common ancestor of gene 3. They're separate from one another. They're not together in a common ancestor. Now, if you trace them even farther back to the chicken, they're still separate, but all the way back to the ancestor of both of these, and you end up with a gene duplication event. And so that's the common ancestral gene. But if it is a paralogous gene, they trace back to a gene duplication event and not where the species broke up. So that's the easiest way to remember and distinguish, let me go back, orthology and paralogy. Orthologous genes trace back to a single gene when the species break up. paralogous genes don't trace back to a single gene there. It could occur before that or after that, but somewhere there was a gene duplication event, and that's where those two genes get together. And so paralogous genes originate from duplication events, and organisms can carry multiple copies of homologous genes. So alpha hemoglobin and beta hemoglobin are just two examples of those. Another really great example are the Hox genes. So when genes first duplicate, they're located right next to each other. Most of the time. There are some rare events that could move one around and make a copy somewhere else, but most of the time they're still right next to each other. And in the Hox genes, they can be lined up and we can find their exact same conserved areas, and some areas have diverged and are different. But the fact that they're located next to each other and that they have these conserved sequences means that they resulted from a duplication event of an ancestral gene. And so multiple genes, even in the same organism, but even if we compare different ones, that go back and trace their ancestry to a duplication event are said to be paralogous. Now I'm going to add one more here. We won't get into a lot of detail about this. This is much more important in bacteria and archaea. And xenology is a gene that traces its similarity back to a gene horizontal transfer event. So if I have a gene, let's say that a virus picked up, and this can happen, but it's rare in eukaryotes. A virus picked up a chicken gene. When it infected a chicken, it, instead of picking up viral DNA and being made like a normal virus, it accidentally picks up a chicken gene, and that somehow gets inserted into the germline of a monkey. These are a lot of very unusual events, so for all of them to occur is very rare. But that gets inserted into the genome of a monkey, and then the genome is carrying that gene that is most closely related to a chicken gene rather than to other primates. So that jump over xenologous. Now, xenology, the root word for this is alien or different species, so it comes from an alien species. And xenology is an important force in the evolution of prokaryotes, but not really in eukaryotes. It's actually more important than we thought, but still not a really major function. So just know all three of these, but especially orthology and paralogy, and that paralogy results from these gene duplication events. So if we compare the Hox genes and line them up, you can see these remarkably conserved, and at the amino acid level it's even more conserved. But these remarkably conserved motifs, these are the eight Hox genes from a fruit fly, labial at the top, proboscipedia, all lined up, showing this ancestral motif that's been changed some in these genes, but still similar enough that we can know that they all came from the same gene. So both their proximity next to one another in the genome and this conserved genetic similarity allows us to know that they are all resulted from a single ancestral gene that's duplicated many times to give us 8 different copies, but duplicated to create this. Now, these are said to be paralogs of one another, and if we trace them back and map them onto phylogenies, we could figure out where they originated and where those duplications occurred in the ancestors of different species. Now let's start thinking about this. What's the advantage of gene duplication? Well, an immediate advantage would simply be some amount of redundancy. You basically have an extra copy, and that can be useful in some cases. If we need to make a whole lot of something and we can transcribe 2 copies instead of 1, we might have an extra boost in the amount we can make. Now that's usually not a critical factor, but occasionally might make a difference, so you've got an extra copy. But another thing is it creates variety, right, that evolution can work on. And so as mutations occur, if you've got 2 copies of a gene, we can maintain the original function, but the other one could mutate and change and maybe even take on a brand new function. We maintain original ancestral function, but also gain new things also. So that redundancy provides a little bit of kind of insurance, but then also allows for diversification without losing what you already have. Okay, so those are 2 advantages that you should be aware of gene duplication events, and we see both of those. So these are great examples. The earliest animals had just a small Hox cluster. They didn't have very many Hox genes, and they can't pattern very many different body regions, and so they remain relatively simple. But in the bilaterian animals, this includes arthropods, invertebrates, and echinoderms and mollusks, all the really kind of big complex animals, they have this incredible diversity of Hox genes, which has allowed them then to have new functions for those genes, doing new things, building new body parts, and being more complex. So Hox genes really are one of the major, probably the major genetic reason why bilaterian animals are so complex and can have so many different variety of body regions. Now, when we have paralogs, right, that's two genes that have come about from a gene duplication event, there are three different end games or end results for those paralogs. One of them is usually going to maintain some amount of ancestral function, at least for a time. Maybe eventually it will diverge. But the three basic outcomes are one of them becomes a pseudogene. This is probably by far the most common outcome when we have a duplicated gene. So think about this. You have duplicated gene, they're redundant, they can both do the original ancestral gene, then one of them gets a random mutation that makes it do something else. And we've talked about this. If you have a random mutation that changes the function of a gene, what's the most likely outcome? Now, if it's changing a gene, especially an important gene, the most likely outcome is that gene's no longer functional, it's doing something that's not useful. But if you still have the original one doing the useful thing, that doesn't really matter. And so those organisms reproduce, they can pass on their genes, genetic drift, other things can take over. So what will often happen after a duplication event occurs is you'll get subsequent mutations that make one of the paralogs a pseudogene. Now, pseudogene is just a DNA sequence that can still be recognized because of its similarity to the original gene. So we still recognize it as part of that gene family, but it is no longer functional. It may not be transcribed, or if it is transcribed, it may not be translated or translated into anything functional. But at some level, it doesn't work as a gene, but it's still recognizable because of its sequence similarity. And again, that's almost certainly the most common outcome from duplicated genes. However, rarely, and this is, even though it happens rarely, these ones are kept and become a really critical and important part of animals. So over many millions of generations and after much natural selection, these are the ones that we really see that made the big differences in why animals look different. One is subfunctionalization. So if we duplicate a gene that is pleiotropic, that means it has more than one job, and we duplicate it, then one of those copies could gather mutations, random mutations occur that could make it better at doing one of the functions, and so it would basically specialize in that sub-function of the original pleiotropic gene. And the other one then may mutate and change as random mutations are then pushed by natural selection to become a little bit better and more efficient in that other job. So it frees up some of the constraint that is imposed by pleiotropy, and so we might get subfunctionalization. The final one, as the name implies, this is a fairly intuitive name, is neo-functionalization. And this is really what we're looking for when we're looking for new developmental functions, a gene that can do something new. It might have had an old job, but it mutates, changes, and every once in a while one of those changes allows that new duplicated protein to do something that it could never do before. And so then we get brand new basically inventions, right? A new gene that can do something brand new, and if that's a benefit, it will be selected for, and we'll see that perpetuated in that group of organisms. So gene duplication coupled with neo-functionalization explain the Hox cluster, why we see all those Hox genes together and how one can build the head and one can build the thorax, and all of those together make it a really great model for understanding the evolution of organisms and where all of this diversity comes from. And you can see this is especially important in the vertebrates, where not only do we get many extra Hox genes, but we get entire clusters. So the urochordates have a very, very simple Hox gene. The cephalochordate has one cluster, several genes in a row. But amongst the fish and the vertebrates, we have multiple clusters, which gives us more and more and more potential and more variety and diversity, and explains especially the neurological diversity, but some of the other morphological diversity in the higher vertebrates. It can be traced back to these Hox genes. So we're going to wrap up with an example and then kind of a revision of, remember, our initial hypothesis. So here's just one example. There is a duplicated RNase in some species of monkeys that allow that gene to do something completely different. So one of these has actually taken on a function that allows them to digest cellulose, which most primates can't do. That's something we typically think of as cows, horses, ruminants to some extent, and in other groups some can do this too, but they can digest cellulose, but primates cannot, except in this small group. And we can trace it back to this single gene duplication event followed by subsequent mutations that allowed one of these enzymes to take on additional functions. So not only can they eat other things, but they can, in addition, digest cellulose. Okay, so here's a revision. We now know that all development comes down to this basic set of toolkit genes, developmental genes, of which the Hox genes are a really important one in animals. And when we see changes, like, how do you get a long beak from a bird? It's changes that impact these toolkit genes. Okay, and so we call this regulatory evolution. And this has led to this, oops, I'm going to write it here. I thought I had it down. This has led to this revised hypothesis. So I'm going to just put a question here and then we'll answer it. What is the new revised hypothesis for Evo-Devo? Okay, let's talk about that now. So the original hypothesis, remember, was that different genes would be the large explanation for why a mouse looks different than a chicken. Or you pick any two organisms, right? And although that does occur, there are different genes. By and large, most of these toolkit genes, let's pull that down a little bit, it's going to bother me here. Most of these toolkit genes are very, very conserved. We talked about the one for eye development, that despite hundreds and hundreds of millions of years of evolution, the fruit fly and the mouse have genes that can essentially be swapped out for one another and still have everything develop normally. So these toolkit genes are way, way more conserved than we would think. And this also explains some of the reason why you've heard this before, that you're 99% genetically similar to a chimpanzee and 80% similar to a mouse, because there's a huge amount of conservation in developmental genes, other genes too. And so even though there are some changes, there are some new genes that can explain some of this, the vast majority of genetic differences, especially in the animals, but across all life on Earth, is explained more by how those genes are regulated, how they're turned on and off. So a mouse has the same basic, with a few minor modifications, but the same basic toolkit as a human. And we can look at homologous and paralogous copies and find most of them. If you find a gene in a mouse, it almost always has a homologous copy in a human. And so it's more about how you regulate, use, where and when you turn on certain genes during development. It's about how you use the genes and not as much about what genes you have. So that is the new revised hypothesis, that regulatory evolution is really the primary, most important thing. And genes can maintain huge amounts of similarity and conservation over many, many hundreds of millions of years, but just be used at different times, in different amounts, in different places, and that leads to dramatic differences downstream. All right, that wraps up Evo-Devo.